My name is Freetong Wakora. I am the Program Manager of Support Services here at IDF. And would like to welcome you to the session for Nuclear Factor Kappa B Essential Modulator Deficiency, also known as NEMO. At this moment, I'm going to share a disclaimer. IDF, Immunodeficiency Foundation, Education Events offers a wide array of educational presentations, including presentations developed by healthcare and life management professionals, invited to serve as presenters. The views and opinions expressed by guest speakers do not necessarily reflect the views and opinions of IDF. The information presented during this event is not medical advice, nor is it intended to be a substitute for medical advice, diagnoses, or treatment. Always seek the advice of a physician or other qualified healthcare providers with questions concerning a medical condition. Never disregard professional medical advice, nor delay seeking it based on information presented during this event. At this time, please join me in welcoming our presenter, Dr. Eric P. Hanson from Indiana University School of Medicine and Riley Hospital for Children. Welcome, Dr. Hanson.
Thank you. And hello to everyone who is watching. I look forward to talking with you in about 20 minutes. I am going to be talking about the primary immunodeficiency known as NEMO Syndrome, one that has some significant features of immune dysregulation in some individuals. So disclosures are that my spouse works for Eli Lilly, and we have stock options, and I'm going to talk about some signaling, some disease aspects, some therapies that are used, and diagnosis. And the focus today of what we're going to talk about is with the thought to precision diagnosis and precision management. And so, when I think about that, it seems particularly germane in the case of NEMO syndrome because of the just real impressive variability in terms of disease phenotypes. This means that individuals can have all kinds of different, difficulties with their particular NEMO syndrome. And so it definitely is not one size fits all in terms of diagnosis, in terms of management, and so we'll touch on aspects of that. Just a little bit about me first, right? I'm at Riley Hospital for Children in Indiana, which is shown here in the state of Indiana on the left, and we see patients from all over the state and bordering, and it's a very Riley Hospital is a very prominent children's hospital in the region. I've been here for the last 2 years, was at NIH in Bethesda, Maryland for about 10 years before then. And prior to that, did my rheumatology fellowship training and research at Children's Hospital of Philadelphia in the lab of Jordan Orange, is where I actually first came across NEMO. And so most of what I know, is based on, what I learned from him and started in that lab, and then what I've picked up in my time at NIH, where I've really been focused on this gene and this protein, but then other parts of the immune system that are impacted by NEMO and NF-kappa B. And so we see patients in my immunogenetics clinic and are able to evaluate individuals who may have primary immunologists or rheumatologists outside of the state, and they might not be from Indiana, but we're able to give input and help guide some decisions through some specialized studies that we do. So about NEMO syndrome, again, it's highly variable. This is a disease that arises due to genetic mutation in a gene that has the name of IKBKG. So, IKBKG doesn't flow so easy. So, it's easier to say, NEMO syndrome. But technically, if you get diagnosed with this, it would be because of some lab studies that were done and some physical features that the patient might have, and it would be due to a genetic finding of a mutation. And usually, when we're talking about mutations, we're talking about the gene mutation that leads to a protein mutation. And it's really the function of the protein usually that we're focused on when we're thinking about how a mutation or a lack of proper function would affect the immune system and the patient. So NEMO NEMO syndrome is known as EDA-ID, and that stands for ectodermal dysplasia with anhidrosis and immunodeficiency. So we know now for decades, 15 years, that there are patients with deletion of NEMO or mutation of NEMO that don't have ectodermal dysplasia. So I'll tell you what ectodermal dysplasia is in a sec. And there's also some patients with NEMO mutation that and have disease, but they don't have immunodeficiency, or at least they might have some signs that could suggest immunodeficiency. They might have some lab findings that would show that the immune cells aren't functioning 100% right, but they might not have had any severe infections or recurrent infections or unusual infections. So, it's the more we know, the more we learn that there's a lot of variability with the NEMO. But many, in fact, most patients that we think, most ones that we know about, have ectodermal dysplasia going along with their NEMO mutation. And so this is something that is shown by abnormal hair follicle development. Patients can have a lack or lower numbers of sweat glands than normal, and their teeth might not develop normally, so that they would have either missing teeth or teeth that are cone-shaped, and in some cases have bad problems with cavities. And in addition, some of these individuals can have some very prominent foreheads. It's called frontal bossing is how that's referred to. As far as infections that individuals with NEMO mutation might get, there are ones with are called atypical mycobacteria. And so these are not tuberculosis, not the same mycobacterium that causes TB, but these are microbes that are found in the soil, on plants, just everywhere, and don't usually cause a problem to people with an intact immune system. These infections with mycobacteria, the atypical mycobacteria, can infect the skin. They can infect the bone. They can spread to other organs, the liver. They really can go to many places, and it can be very difficult to clear once an individual with a NEMO mutation and immunodeficiency is infected. But usually, very prolonged courses of antibiotics managed by infectious disease experts are given and can clear the mycobacteria. Another class of infections that people get are with encapsulated bacteria, and so these are ones that antibodies usually help prevent, and these would be sinusitis or pneumonia, and essentially getting a replacement immunoglobulin, really makes this problem, much less of a problem, much less likely to occur. Herpes viruses can also affect individuals with NEMO mutation. They can infect, different parts, and then opportunistic infections. So a classic one would be something called pneumocystis that can cause a pneumonia. And the thing is that some people with the NEMO mutation might not have much of a risk of this because the one part of their immune system that is the one that kind of handles and prevents opportunistic pneumocystis would be okay generally. But other individuals with NEMO mutation might have very low CD4 T cell counts that would predispose them to this. And so, talking about precision medicine, we're not yet at the cutting edge level of the science and medicine to be able to really confidently predict who would and wouldn't be able to get infected with an opportunistic organism. So most practitioners would prophylax their patients with NEMO mutation who has shown some aspect of increased risk of infection. But down the road, that will be the kind of thing that we might do, that we would tailor someone's immunoprophylaxis, based on their specific T cell and B cell and other immune cell, not only number, but their function of these cells. So there's a very broad spectrum. One thing that in my own career I've focused on more is the immune dysregulation part of it, the inflammatory disease component. And this is something that can appear in individuals who had a lot of infections, but also in some that haven't. So we earlier on noticed that there seemed to be two parts of the protein where you'd see more of these inflammatory disease features. So one of these is that so this is the NEMO protein here in gray, and just happen to have marked some of the domains that are known to be important for different signaling with other cells. But basically, we noticed that there was two, broadly speaking, two domains where more of these inflammatory diseases, had appeared, and these can be of all kinds. Any of the itises, I like to think of it as vasculitis, arthritis, paniculitis, which is inflammation of the fat cells, lead to nodules under the skin, but neuritis, uveitis, which is inflammatory eye disease, colitis, and so really some of our earlier work was looking at these ones that lead to the very end of the protein being absent. And we noticed that those mutations are shown here in red. So here's the same NEMO protein. And basically, we're just listing here, going from top to bottom, the mutations that appear at the very end of the protein. And most of the ones at the very end cause a single amino acid change in the protein. But ones that are in this box are ones around here. And those actually lead to a change so that there's a stop signal to the protein. Proteins get made from the N terminus to the C terminus, so from left to right. So if the protein's getting made and then there's a signal to stop, that protein will be shorter and it will lack this important domain at the very end. So anyway, these individuals with these particular mutations have severe inflammatory disease. And the thing that we were struck by was that some of their sisters and mothers and aunts also have diagnosis of a severe disease. And this is something that looks very similar to a disease called Behçet's disease, where there could be oral ulcers and genital ulcers, and there can be problems with blood clots, either in the head, in the brain, or lungs, other parts. There can be also colitis, and so this is different than some of the other cases where there's females affected by a NEMO mutation, but those females can't have any male offspring because the mutation is too severe. So this is something a little bit in between, but that's something that as we study NEMO syndrome more and the disease more, but there are more and more females that are getting diagnosed. So this is a little technical, and we're going to skip over it. But I skip over some of the details. But I did want to just sort of show, getting into sort of some of the precision aspects of this, some of what we do, and this is stuff that actually is not, to my knowledge, done by any clinical labs, but are done on a research basis. But the information that we get from this can impact how we manage patients clinically. So what I'm showing you here is a list. Every row in this series here corresponds to a different gene. And so now we have about 20,000 different genes in our body. And NEMO actually controls this other protein, group of proteins called NF-kappa B. And what NF-kappa B does is, when it's activated, it leads to genes, particular subset of the 20,000 genes that we have. It leads to those genes either being produced in greater amount, meaning the instructions that they contain are read and lead to more of those proteins encoded by those genes being present in the cell. And in some cases, NF-kappa B activation leads to some genes being made less. And so what we're showing here are on the along the top, we're saying NDAS, patient 3, patient 1, patient 2. And so some of these are repeated gene expression results. So this is gene expression data where we're comparing to 13 healthy controls. So these are individuals without an NEMO mutation, and we kind of compressed all of their data into one column. But by definition, we said that those healthy controls have normal levels of these genes, normal levels of expression of these genes, I should say. So it's not and then in comparison, these patients with this subset of NEMO syndrome that we're calling NDAS because of how the protein is formed abnormally by deleting it, the part that's coded by exon 5. But anyway, you can see that these individuals have some of their genes are a lot lower than normal, and some of the genes, expression of their genes, is a lot higher. And so the last three columns we're comparing to an autoimmune disease called systemic lupus, and it looks pretty similar to that. So definitely not a typical thing for an immune deficiency. And so, but the point to make here is that we're able to by getting a blood draw from an individual, know that they have this profile and this profile that is similar to another disease. And so the point is that if we know what this other disease might respond to, what medicines it might respond to, or what risks those individuals might develop, it definitely leads us to think about the possibility of using some of the similar diagnostic tests or medications if we're looking for a treatment for a certain disease phenotype, and so blocking this interferon signature is something that has a rational basis, and so this is something that would be done on an individual basis. So it'd be a very different pattern and signature for these individuals compared to other individuals with a NEMO mutation that doesn't have the same subset of disease features. So I think that this is helpful for some people to know, but basically that the NEMO protein is inside the cell. It interacts with a lot of different other proteins, and there's a lot of different immune receptors that need to be able to use NEMO to signal and send the NF-kappa B into the nucleus. And so again, this is what I was kind of talking about with the previous slide, that when that NF-kappa B goes into the nucleus, it binds only certain genes and leads to those genes either to be read into more protein or those gene mRNAs to be lower, so that there's less of that protein around. And so that's how the cell responds. And now some of the things that are important to think about in terms of making a diagnosis. So when we're testing whether NEMO is functioning properly or not, there's different ways of doing that. One of them is to test for when we stimulate cells that would have the mutant form of NEMO, whether when we activate this receptor, do we see any NF-kappa B go into the nucleus? Because when there's a defect in NEMO, then it would impair the ability of this event happening. And so, that would be one particular kind of test that we would do. Other things that could be done would be to measure some of these proteins that get made once the NF-kappa B goes into the nucleus. We can see that released from a cell. And some of these are clinical lab tests that can be ordered from any hospital that we can measure these cytokines in the serum. And so usually that's called a cytokine panel. And so on occasion, these can be followed, and they can help us know more about an individual person's NEMO type. And so now the other part of the diagnosis is, so that I was talking about a little bit about NEMO function that we test for. The other one, getting before that, would be the mutation in NEMO. Detecting the actual change in the genome that would lead to a mutant NEMO protein getting made. So, one of the things that many of you will be familiar with is something called whole exome sequencing, and that's different than whole genome sequencing, and that's different than something else called Sanger sequencing. And so, what I'm showing you here is the NEMO gene, and that's the part I'm sort of showing with my mouse. And the NEMO gene is made of these blocks and these black lines. And both the blocks and the black lines are just a string of what we call base pairs. That's DNA. And so the thing is that the blocks code for protein, and the intervening sequence has other functions, but they don't end up having the code for the actual NEMO protein. So when you get whole exome sequencing done, or someone does that, we're determining the sequence of these blocks. And when you get whole genome sequencing done, the sequence of the entire gene is done, and not only the NEMO gene, but of every gene that you might have, because this is done when an attempt is being made at finding what the mutation could be that's causing problems. So Sanger sequencing is different. That's done when somebody has a patient and they have features that are really classic or convincing of NEMO deficiency. And so then they might then do sequencing only of the NEMO gene, and would typically only do these exons of the NEMO gene. And if a mutation is found, then that would lead to maybe some other testing to confirm that, and then things go on. Now, the one thing that makes it tricky is that there's this thing called a pseudogene. And so a lot of times with whole genome or whole exome sequencing, it's hard for the algorithms that we typically have to know whether the sequence came from the NEMO gene or this pseudogene, because the pseudogene's sequence is very, very similar to the NEMO gene. And so basically, some special things have to be done sometimes to be able to, be sure that an individual has a NEMO mutation. Some of these things are, doing something called long-range PCR. So we don't just sequence the individual blocks, we sequence from one end all the way to the other end in one go. And that way we can be sure that we've got the sequence of the NEMO gene, not the pseudogene. And then there's other things that are being done, and there's a lot of people working on ways to make this easier because of this problem. So this is just to illustrate. This is another gene that's important in this same pathway. So you might remember I'd shown that NEMO is important when an immune receptor gets triggered. NEMO is important so that this inhibitor of NF-kappa B can get degraded, and that's what lets NF-kappa B go into the nucleus. Anyway, this inhibitor, people have mutations in that too, and that can cause a disease that can look a lot like NEMO syndrome. But anyway, this is just showing all of the gene sequencing results from over 100,000 people that is a publicly available database. And you can see that the exome sequencing is shown in blue and the genome sequencing is shown in green. And you can see there's what we call good coverage of all of the exons. So, all of the data submitted shows us that really, for all of, every individual sample that's sequenced, we really know about this I-kappa B alpha sequence. But in contrast, this is the NEMO gene sequence, and you can see that the coverage is just very poor. And again, the reason is because when sequencing is done, in a lot of cases, it's not possible to assign the sequence that is obtained to the pseudogene or to the NEMO gene, and so it's not put anywhere since it can't be done unambiguously. So that's why the specialized techniques are sometimes needed. And so just going to talk a little bit about COVID. It's on people's minds, certainly, and how it might impact things. And I'm just pulling up, just literature that I had found. And so this is about an individual who had received a stem cell transplant for his NEMO deficiency, and then got a COVID infection. And he was treated with a viral inhibitor and high dose intravenous immunoglobulin, and got some additional treatment, convalescent plasma that would contain antibodies to the virus that would help reduce the ability of that virus to propagate in the body. And he did well, although he was having some lung involvement that was worrisome. So I just want to mention some work that's been done in the past couple of years that a very large group put together a lot of sequencing data from patients that had severe COVID-19 infection. And they had predicted that based on what we know, that these individuals would have mutations in these different genes. And one of them is NEMO, because it is known that NEMO is important for the antiviral response. And so actually they found that there were mutations in these three genes, TLR3, IRF7, IFNAR1, not NEMO, but it is a concern. So we would think that most likely an individual with the NEMO mutation would have a tougher time with a COVID infection than someone else. So we happen to be doing some work on that ourselves because the actual COVID virus attacks NEMO in a way. It cleaves NEMO into parts, and so we're interested in seeing what we can learn about how what we understand about that process has to do with the disease that we're seeing. So other reports from recent literature, and then I want to make sure we have lots of time for discussion, so we'll go through faster. But having to do with transplantation, that's a big, somewhat open question that we just keep getting more information on yearly. But there are studies from 5 years back, including many more patients. It's a very rare disease, so 30 patients is a lot of patients. But that it appears that reduced intensity conditioning with transplant works well for NEMO. And one other thing to say from just previous work, that we have found that individuals with current mycobacterial infection at the time of transplant, or ones with colitis, would tend to do less well than individuals without those. And that was work done by Capuchin Picard and with others, and came out about 5 years ago. And so, one thing that is really important is to really, prevent and eradicate those atypical mycobacterial infections, aggressively, because they can just become intractable and really be a problem. So other report that came out in the past year, this one describes an individual that actually, after transplantation, the transplant took fully, but then it looks like ended up being a mix of the patient's original bone marrow and cells and the donor. And this individual had really bad gastrointestinal problems. One thing that made this better, and these gastrointestinal problems were so bad that what appeared to be happening was the bacteria from inside the intestine was basically leaking into the bloodstream from there. So when this individual was put on regular courses of really antibiotics that would clear these bacteria, they really stopped having these problems. So that would be something where we're not there yet to really understand exactly what to do, but in general, learning more about the microbiome and how that affects things. But certainly, if gut issues and motility and bacterial overgrowth are a problem. And certainly if bacterial bloodstream infections are a problem, this would be a particular something to consider. Lastly, about the colon, we understand a little bit more about this, all the time, but that basically, even though someone can be immune deficient, they might sometimes need immune suppression. And in particular, disease features in which we think, cell death is a factor. And with NEMO deficiency, that would lead to infections. We also know that there can be cell death of, for instance, the intestinal epithelia. And this is something that's blocked by blocking one of the cytokines called TNF that does this. So this individual, after their transplant, had problems with this, and then actually looked like their colitis got worse after transplant, but then did respond with blockade of TNF. And so here's describing a little bit about that. It's not just the bone marrow cells that use NEMO and need NEMO. Sometimes it's other cells. This is showing the gut epithelia. So here's the bacteria inside the intestine. Here's the body, the tissue. So when there's cell death here, these little microbes can go through. And after transplant, sometimes there can be a need for even more immune suppression, because now this immune system is more active because it doesn't have the NEMO mutation anymore. So anyway, this would be something that wouldn't be the case for certain individuals with NEMO mutation who might also have infections and problems with mycobacteria, but they might not have a problem with TNF-induced cell death. And so this is would be an example where knowing the specific mutation that an individual has and how it functions really would guide what you would think of doing in terms of treating that disease aspect. So this went a little bit longer than I wanted, so I will first off say that I think my info was in one of those early slides. If anyone wants to reach me after, they can contact me and we can set up a time to talk. I'm always happy to do that. And let me just get that on there. So thanks very much, and I'm happy to take questions.
Thank you so much, Dr. Hanson. Okay, we have one question. What are some of the herpes viruses, such as flat warts? Have you seen any treatment options been successful? With some of the herpes viruses, such as flat warts, have you seen any treatment options been successful?
So no, not that there haven't been, but so I'm sorry, and then maybe we can back and forth with the chat. But so is it flat warts? Is that what it was?
Yes, flat warts.
Yeah. So no, I don't know, but I think that this is the kind of thing where there are individuals that are specialists in sort of antivirals and other therapies, and warts in particular, and the whole kind of immune response involved around that. So I think that in the theme of precision medicine, I might just say I can't tell you from experience a specific therapy, but I could sort of tell you an approach that one might take. There certainly, you would reach out to, through your immunologist, or, you could, I could look into it. But basically, one would be able to really kind of try to understand the particular NEMO mutation. And, there might be some things that we know already about that that mutation and how certain immune cells might be defective. And it could be that we have an idea that it's the skin cells that are the problem mainly, or we could think that it's the immune cells, circulating in the blood that are the problem, just based on that mutation and potentially some additional tests that would be done in the lab. So if then knowing that, could lead one to make a choice on something to try that might not have been thought of. But I think that, working with sort of a research lab potentially, or an expert dermatologist that deals with warts and sort of is open to considering maybe therapies that wouldn't typically work for somebody with warts but would work for somebody with a NEMO mutation in warts would be a thing to try. So, but I do feel like I want to get back to you with a better answer because someone might know this and I could probably look into it. So email me if you want so I can get back to you or figure something else out.
Can you clarify the COVID sequencing study? Did they not find NEMO in the study, but they still think NEMO patients will respond poorly to COVID?
Sure. So let me pull that up again. So I looked through this study and let's see here. Can I advance to that slide? From what I saw, they didn't find NEMO. I think that that doesn't necessarily mean that someone with a NEMO mutation wouldn't be at increased risk. The thing is that, as I had mentioned, sequencing the NEMO and being sure that it's truly a NEMO mutation can be tricky, and so I'd have to look at that there's a possibility that some sequencing would miss some, and some of these individuals with severe COVID outcomes might have had a NEMO mutation. Meaning that it just wasn't detected, but because of some inherent difficulties of detection, not because they weren't there. But what we do know is that in patients with either, this is called toll-like receptor 3, the receptor that recognizes the virus's nucleic acid, that patients with a mutation in that can have a severe viral infection. And same thing goes for, it's been shown, patients with NEMO mutation. So this is, this sort of list of genes, this schematic that shows the genes that were thought by these experts to be the ones that would be most likely to lead you to have the highest risk. That's based on sort of general understanding of viral infection and viral immune responses, the antiviral immune responses, what's important for that, NEMO being one. So hope that clarifies. Now some of our work is looking at the sort of the flip side of that, but that might not be too relevant for most people. But in some very rare cases, it could be that some NEMO mutations would even make you less susceptible than somebody even without a NEMO mutation at all. It's possible, but generally, if there's immune deficiency, we do think that there is risk.
Okay, so I don't see any more questions coming in. So thank you so much to our listeners for your phenomenal questions. And again, Dr. Hanson, I want to say thank you for this very informative presentation.